This is Dr. Guru Sampavde from the Advent Health Cancer Institute presenting Abstract 546, or Neural Network Analysis of Tumor and Germline Profiling to Predict Survival of Muscle-Invasive Bladder Cancer Following Radical Cystectomy, an analysis of the Cancer Genome Atlas, or TCGA. I will present the initial section of this presentation, and then Lane Sadler from AIQC, or Artificial Intelligence Quality Control, will present the remaining segment. The background is that following radical cystectomy for muscle-invasive bladder cancer, recurrence is suboptimally associated with pathologic stage, tumor mutation burden, or TMB, an apobic mutational signature, and minimal residual disease, or MRD assessment using ctDNA are all associated with tumor recurrence and/or survival following radical cystectomy. We hypothesize that molecular information from tumor and host tissue may capture orthogonal information and provide comprehensive prognostic information to help select patients for adjuvant therapy. So the objective of this study was to propose a prognostic model to predict 5-year survival, employing deep learning to incorporate information from comprehensive genomic and transcriptomic data from both germline and tumor tissue in patients with muscle-invasive bladder cancer undergoing radical cystectomy. Now, our major collaborator and colleague, Lane Sadler, will present the design methods and results and conclusion of this study.
Thank you, Dr. Sampavde. Turning our attention to the methods section, we'll see that TCGA had already filtered out neoadjuvant chemotherapy patients as part of their original protocol. However, they did not provide researchers access to germline variants, so we called those ourselves using GATK4's haplotype caller and QC'd for depth. We also filtered out common variants above 2% allele frequency using the Broad's NOMAD. And critically, we used Ensemble's variant effect predictor to capture high and moderate functional impact variants with respect to how they influence the protein. Now, when it comes to survival, we used 5 years as the overall landmark, meaning that you either had to have died within 5 years or survived beyond 5 years with continued clinical follow-up data. Patients that died within 4 months of radical cystectomy were excluded in order to avoid surgical complications. And then at the opposite side of the spectrum, if you died after 70 years, you were a statistical outlier, and therefore you were ruled as a survivor. As the cohort was imbalanced toward dead patients, we propensity matched 39 survivors against 2 sets of 39 dead patients for a total study of 117 patients. The factors used for matching included pathological stage, adjuvant chemotherapy, and other demographics. The samples were divided into 3 equally distributed subsets of alive and dead patients. The algorithm was trained on 50%, evaluated on 40%, and tested on 10% of the dataset. Remember, we've got those additional 39 holdout samples for added validation as well. The features that are fed to the linear neural network are aggregated at the gene level. So for expression, we have normalized gene expression quantification counts. And for variants, all of those functional variants are grouped at the gene level. The accuracy of the algorithm was 97% during training and 95% accurate across the 79 evaluation samples. Now, when we permute these features and observe the impact that their shuffling has on the model's baseline loss, what we observe is very interesting. 19 of the top 20 features are germline genes. 10 of them are involved in immune system pathways, and 6 of them are involved in cell cycle pathways. The reason why these results are so striking is that they challenge the assumption that germline variation in a prognostic and therapeutic setting is unimportant in comparison to tumor-specific omics. To put it lightly, this calls for a reanalysis of many data sets and could provide new weapons in the war against cancer. Thank you.